అమరావతి
సార్
మిమ్మల్ని హోస్ట్ గా చేశాను, మీరు కూడా రికార్డ్ ప్రెస్ చేయండి.
ఓకే సార్. ఓకే సార్.
నేను మళ్ళీ రీక్లెయిమ్ చేస్తాను హోస్ట్.
సార్
మీరు జస్ట్ రికార్డ్ బటన్ ప్రెస్ చేసి వదిలేయండి.
అట్లే సార్. ఓకే. సార్
అమరావతి చెప్పండి.
పర్మిషన్ ఇవ్వలేదు సార్. ఇంకా
ఇచ్చాను ఇంతకుముందు.
ఇంకా రాలేదు సార్.
ఇప్పుడు చూడండి, ప్రెస్ చేసి.
ఇంకా రాలేదు సార్. ప్లీజ్ ఆస్క్ ది హోస్ట్ టు గివ్ యూ పర్మిషన్ టు రికార్డ్ అని వస్తుంది సార్.
ఇక్కడేం చూపించట్లేదు.
మేబీ కొంచెంసేపు పడుతుందేమో, చూద్దాం సార్. వెయిట్ చేద్దాము. మీది రికార్డ్ అవుతుంది, లింక్ వస్తుంది సార్.
అదే నాది రికార్డ్ అవుతుంది.
మీది అవుతుంది. నాకు ఇంకా పర్మిషన్ రాలేదు సార్. మొన్న అయితే వచ్చింది. మొన్న
ఇప్పుడు చేయండి.
స్టిల్ అట్లానే వస్తుంది.
ఓకే, ఎనీవే యూ కెన్ స్టార్ట్. ఇక్కడ రికార్డ్ అవుతుంది కదా.
ఎస్ సార్, మీ సిస్టమ్ లో రికార్డ్ అవుతుంది సార్.
ఓకే.
ఓకే సార్. ఓకే సార్, లెట్ మీ వెయిట్ ఫర్ 3 more minutes sir. Now participants reach 48. 3 minutes, 2:10 I'll start sir. Let us wait for some.
Okay.
Sir, నాకు కూడా పర్మిషన్ వచ్చింది సార్.
ఓకే ఓకే, క్యారీ ఆన్.
Yes, and also recording sir.
Hello.
Yes. Who is this?
Ma'am?
Yes, tell me.
Yes. Ma'am, the video links, YouTube video links which you have shared, so they are not opening, ma'am. It says that video is unavailable. No, none of the videos are opening. I just want to know whether it is only for me or for others also. It is saying that video is unavailable. This
Uh-huh.
video is private.
Yeah, okay. That Dr. VLN is going to answer you.
I think for others it's opening. Initially I sent 3 videos, but later I sent 3 more videos. You check the latest one. See, somebody is
Okay,
saying
sir.
yesterday I could open it. I think I also tested. Just check the latest mail.
Sure, sir. Okay, sir.
Today also I sent Saturday's video.
Sure, sir. I'll check it again.
Yeah.
Thank you.
Okay, Amaravati.
Yes, sir. Okay, dear participants, a warm welcome to this online workshop, Molecular Data Analysis through Bioinformatics Tools. It is a great privilege and honor to welcome Dr. Lakshminarayan Reddy, sir, Associate Head, Department of Molecular Biotechnology, SV Agricultural College. Dr. Reddy obtained his MSc HE degree from Genetics and Plant Breeding, SV Agricultural College, Anglo Tirupati in year 2000 and PhD from Center for DNA Fingerprinting and Diagnostics, Hyderabad in 2008. He did his postdoctoral fellowship from Oklahoma State University, Stillwater, USA in 2014 and the Raman postdoctoral fellowship sponsored by the University Grants Commission in New Delhi. Dr. VLN Reddy sir received many scholarships and awards. Among them, he received Junior Research Fellowship awarded by CSIR between 2002 and 2004, Senior Research Fellowship between 2000 and 2007 awarded by CSIR, Raman Postdoctoral Fellowship awarded by UGC in 2013 and 14, Travel Grant to participate in international workshop on engineered crops held in USA in 2014, Tata Corner Initiative to support for young Indian scholars to attend second international conference on global food security held at Cornell University, USA in 2015. He has more than a decade of proven research experience in rice molecular breeding and genetics, as evident from his publications in reputed international journals. He was involved in development of capillary electrophorese-based microsatellite DNA fingerprinting protocol, which facilitated quick and accurate detection and quantification of adulteration in basmati rice. And thereby, it helps and expand and sustain our export basmati rice export opportunities for India. From his APEDA CDFT, Center for Basmati DNA Analysis has been established at CDFT Hyderabad, which is being certified the export samples from EIA and EIC of Government of India. Dr. Reddy was also involved in identification of several promising QTLs linked to yield and its components from wild species and primitive cultivars of rice. He also mapped QTLs governing important quality traits and dissected rice of basmati rice. He also identified a novel dwarfing gene, that is, alternative dwarfing gene, popularly known as rice. He has also identified evenly distributed hypervariable rice microsatellite markers that has a potential to use for background selection in marker-assisted selection of rice. Further, he disproved the age-old fact that the genetic diversity of modern cultivars is getting narrowed down. Contrary, he demonstrated that the molecular genetic diversity of major Indian rice cultivars over the decadal periods from 1960 to 2000 is increasing. Dr. Reddy developed a DNA fingerprints and also QR codes for major Indian rice cultivars using molecular markers. He uncovered many novel alleles from key yield genes by targeted research. Very recently, in the yield and drought stress, that is DTY4 and DTY 2.2, have been transferred into two popular rice varieties, that is MTU 1010 and NLR 34449, employing marker-assisted backcross breeding. In short, his contributions to rice genetics and breeding is invaluable and most significant for the farming community. Now, I request Dr. Lakshminarayan Reddy sir to start the highly informative session of the workshop, that is phylogenetic and population structure analysis, employing the softwares Mega, DARWIN, and Structure. Over to Dr. Reddy sir.
Thank you, Dr. Amaravati, for a nice introduction. Is my voice visible?
Yes, sir. It is clear and audible. Your screen is also visible.
Okay. Thank you very much. So a very good afternoon, all of you. I welcome you all for the 5th day of our workshop. So today also we have one of the important topics. I think majority of the MSc and PhD students, we can see assessment of genetic diversity or development of the phylogenetic trees as their PhD work. So this is actually a very important topic and very big topic also. Usually for my PG students, I take 2 to 3 theory classes and 2 to 3 practical classes. So covering all the theory and practical in within 3 hours, as you know, it's a difficult thing. But I try to give just basics of theory part, and I would like to focus mostly on practical part as it's a workshop. Okay, so coming to this my topic, that is molecular phylogenetics. So as you know, the organisms on the earth, we have some relation with each and every organism. Like we have a close relationship with monkey or chimpanzee, like insects have other relationship. Like this, all organisms, right from bacteria to human, mankind, we have some association, or we can say some relationship. And if you observe closely, we can classify them into different populations based on their morphology, based on their habits, and based on their DNA and so on. Okay. So this phylogenetic analysis mainly deals with this. So what is the criteria to classify all these organisms and how it is useful? Okay. So I will just give a definition of this one. It's a phylogenetic tree is it's a evolutionary history of living organisms using like a tree-like diagram. As you know, tree has a root, stems, branches, and leaves.
Sir, your screen is not visible, sir. I think that is not showing.
Is it not visible?
No, it is visible, sir.
Visible, sir.
Maybe he has a specific problem. Sir, check your internet.
Yeah, okay, now it is.
Now is it okay?
Okay,
It is
sir.
visible, sir. It is visible.
Okay. So it's like knowing the evolutionary history of all organisms with the help of a tree-like diagram. It's as simple as that. Okay. So if you see the definition of just phylogeny, like a tree branching pattern, we represent a relationship of evolutionary divergence in the form of phylogeny. Okay. So whatever outcome comes out of the phylogeny is phylogenetic tree. Literally, phylogeny means phylon means it's a tribe or race. Geneticos means related to birth or genetics. So as you know, molecular phylogenetics means here we use mostly this molecular data, either molecular markers or sequencing data. Okay. So if you see the importance of this phylogenetic tree, there are so many advantages are there. we can classify all the organisms into different groups or different populations, and it can answer many biological questions. Main important thing, recent days we got benefited from this kind of studies is identification of the pathogen. So, for example, this coronavirus, which was identified in 2019, when it was first identified, immediately we can use this technology like molecular phylogenetics. What is the origin of this phylogeneity? From which organism it was originated, whether it is from other bacteria or viruses, from mammals or whatever. Okay, so it was concluded that this coronavirus was originated from China mostly, and closely related to organisms are pangolin, or you can say bats also. Bats and pangolins, mostly this coronavirus we can find it. So like this, there is a lot of implications are there for this phylogenetic analysis. So I don't want to go into the details. I will go to the actual topic, that is phylogenetic analysis. So in this class, I would like to stress mostly what are the different terms which we use during this phylogeny analysis, and how do we construct a phylogenetic tree. So these two things only I will restrict. Actually, otherwise it is a very huge topic. Okay. So as I told, phylogeny is a branching diagram, which actually shows the relationship between the different species which share a common ancestor. So for example, here you can see there are 4 species are there, A, B, C, D. Here you can see A and B are closely related because they originated from a common ancestor called E. Similarly, this C is closely related with AB because these ABC has common ancestor compared to D. So like this, we can try to identify what are the different closely associated species and what are the distantly related species. Okay. So these are the different commonly used terms during the phylogenetic analysis. So usually dendrogram we use, it's like a broad term. Like dendrogram is nothing but a diagrammatic representation of any tree, phylogenetic tree. So within the dendrogram, there are 3 types are there: cladogram, phylogram, and chronogram. Cladogram is also a tree, and phylogram, these are all trees only. But here, a branch length do not represent time. Cladogram, here you can see in this example, it doesn't have any meaning. Means it will not reflect the closeness or distanceness of a species. Simply, it will say which species are closely related, which species are distantly related. But branch length doesn't have any meaning. But if you see the phylogram, it has a meaning. Branch length indicates actually how distantly or closely related different species. Okay. The third one is the chronogram. As the name indicates, it represents the evolutionary time also through its branch length. So that is the only difference for this cladograms, phylograms, and chronograms. Okay. So some more terms are node, internodes, and root and lineage. Just like a tree, if you just imagine a tree, here you can see the nodes are internodes, which actually connects different species. Like here you can see B and C, they are connected through this node. Okay, and A, B, C are connected through this internal node. Okay, and these species are also called operational taxonomic units, or it's also called taxa. Okay, and these are also called terminal nodes. Okay, and this branch actually shows the distance between different organisms. Like here you can see this is the root, right? This, the first point is root from which all these species have derived. From this root, actually 2 groups have come. A, B, C have separated and D, E, F have separated. Okay, and from this again immediate ancestor, you can see from this immediate ancestor A, B, C, 3 species have derived. Okay, like this DF have derived. Okay, like this we have to remember this branches, roots, nodes. Okay, and this closely related ones like here D and E. This we can call it as a clade, one clade. Here also you can see BC or one clade. Okay, so these are the some of the things. Okay, and this also I was telling this is one clade, this yellow color one, and this blue color one also one clade, this orange color one like this. So another terminology is binary tree. See, if a common ancestor have derived initially B and C, then again B has separated into D and B. That means every time it is divided into two species. That's why it is called binary tree. So other branching pattern here you can see dichotomy. Dichotomy, every time whenever an ancestor derives, it will uh, uh, split into a two species. Okay, and in case of the polytomy, it split into a multiple species or multiple ancestors. Okay, so radiation means it's a multifurcation. Yeah, because of if we don't have any solid data, then our tree will be like this radiation. That means if you do not know the what is the ancestor and what is the distantly related, then in that case you will get this radiation kind of thing. So these things we will see in the practical. So the other one is the unrooted trees and rooted trees. Rooted trees means it has a root that denotes a common ancestor. Here you can see, like a normal tree. Unrooted tree means we do not know what is the common ancestor here. Okay. So another terminology is scaled tree and unscaled tree. So the scaled tree, it has a branch lengths are proportional to the number of nucleotides or amino acid changes. So here you can see one clade, Varaiza, Triticum, and Aragronis. Now here you can see there is no distance at all. That means they are all almost sequence is same. But when compare with the Varaiza and Hardium, here you can or Gia or Saccharum, here you can see the branch is different. That means the number of amino acid nucleotides are different, changes. So this is scaled tree. But unscaled tree, there is no meaning of this amino acid changes. So another classification is monophyletic and paraphyletic. So if a common ancestor derived into two subspecies and each subspecies is very different Saturnite, this indicates only Saturnite part. So other species only Martian part. That is single phyletic, that is monophyletic. But in case of the paraphyletic, here we can find the some they share this species. These two ancestors they share this species also. That is paraphyletic. So here you can see the example monophyletic. Here you can see the organisms belongs to Asteroidae. They all comes under one group, that is monophyly. And Ophiuridae, another species. And like this, you can see a single species has a their own clades or their own subtrees. And paraphyletic groups, we can find the they share the common ancestor. Like here, foxes and the dogs, they share the common ancestor. That's why we cannot draw a clear-cut diagram. Okay, whenever a common ancestors are there. So another important thing is the consensus tree. You know, for drawing trees, there are many methods are there, and many source files we can use it. You know, each source file gives the different types of trees, or each method gives different types of trees. So here, for example, here you can see tree A and tree B. Using the one method, which as per this method, A and B are closely related compared to C. But the tree B, it says A and C are closely related compared to B. So in that case, it leads to a lot of confusion. So in that case, what we can do, we can make a consensus tree. That means we can compromise these A and B trees. We can make a, so we can say finally, yes, A, B, C are closely related. Instead of arguing A and B are related or A and C are related, we can say A, B, C, all three are closely related. Okay, like this, we can make consensus tree. So now coming to the actual thing, how to construct a tree, phylogeny tree. So for construction of the phylogeny tree, what we need is a information. Means what are the different okay. So what are the different traits we can take? Here, always there is a confusion. Okay, because as our participants are from diverse group, humans, what type of characters you have to take, plant people, what type of characters, and microbes like this. So initially, they used to take paleontological records. Okay, so and then followed by the morphology like skull type or skull size or presence of some part, something like that. Okay, then after characters, even behavior also we can take it as a one trait. Like somebody will get up at morning and somebody will sleep night, some behavior. Okay, these also can be used as a character to draw the phylogenetic analysis. But as you know, these traits, mostly the morphological and behavior based characters, they are highly environmental influenced. Okay, so nowadays people are using chromosome based or means number of the chromosomes and the structure of the chromosomes and gene frequencies. Like this, these characters also we can use to draw the phylogeny. Okay. Then now the molecular tools have come, especially the molecular markers and sequencing based NGS technologies, and all these things almost replaced with this morphological based character. But here one thing we have to remember, whatever characters we are taking, phenotypic characters, that will not applicable for microbes, because it's very difficult to observe the morphology of the microbe. So in that situation, we have to go for molecular analysis one. Okay. Now again debate comes, which one is the best, whether morphology based or molecular based. So always phylogenetically, you better to take as many characters as possible, whether it is morphology based or molecular based. If you take more number of characters, we will reach the precise phylogeny, right? So here you can see interesting thing, the increasing number of the publications towards the molecular markers or sequencing based phylogeny analysis compared to morphological markers. Okay. Now coming to the methodology, how to construct a phylogenetic tree. There are actually 4 methods are there. First thing is choosing of the molecular marker and then followed by the multiple sequence alignment. Then we have to use some model to build a tree. And finally, we have to test whether this tree is correct or not. Okay. So first one is the molecular marker. There are n number of molecular markers are there. DNA based markers, protein based markers, like right. So here also many types are there. So here we have to remember one thing. For studying the very closely related species, better to take nucleotide sequences. Nucleotide sequences means DNA based compared to protein, because DNA based markers always they evolve rapidly compared to protein based sequences. Because as you know, the wobble hypothesis, even the whatever variations are there in the DNA, all will not reflect in the protein sequence, because protein sequences are only non-synonymous SNPs variations only will reflect in the amino acid changes. So, because if you take very closely related species, there is hardly any difference in the protein profiling, right? In that case, better to go for DNA-based method. Okay. Suppose if you are taking very widely divergent groups of organisms, then better to go for protein sequence. In that case, we can expect some variation at protein level, or you can say at ribosomal RNA level. Okay. So always, but if you have a feasibility or if you have a facility, better to go for protein sequence compared to nucleated sequence. But the thing is, working with DNA or nucleated sequence is very easy. So everybody uses this molecular marker. Okay. So here I just want to give a classification. I don't want to go into the details, as you already know different types of molecular markers are available. In all types of molecular markers, how we can develop as a molecular marker, we should find a variation. Okay, that variation can come at any point of the chromosome. Okay, so it can be the variation we can see either in microsatellite repeat or in the flanking sequences. Anywhere we can find this repeat variation. So for example, I'll show here simple sequence repeats, how the variation comes. In the allele 1, some 10 repeats will be there. Allele 2, 12 repeats will be there. Allele 3, some 14 repeats will be there. So here variation is there. When you design a primer flanking this region and run in agrogelar proportion, we can see it as a variation, because the small one will move fast and the big one will move slowly. That gives us a polymorphism. So this is actually basic principle. And in case of the SNPs also, these are the most sought after molecular markers. So one single nucleotide change, instead of A, if an organism has a C or G or T, this we call it as a SNP, single nucleotide polymorphism. So like this, either repeat variation or nucleotide variation or sequence variation, anything can be used as a molecular marker. Okay, so like this we can use it. So that is the first step. The second step, multiple sequence alignment. So in the first class or second class, somebody is asking how to identify the SNP, right? So if you want to identify any SNPs or indels, you have to do is sequence alignment. Suppose if you have a 5 sequences here, you have to use either Cluster W or any software or Cluster Omega, which already demonstrated in the last classes. Here you can see the nucleotide variation. Okay, so some species has a G and other species has A in the same place. Like this, we can find the variation. This we call it as a SNP. If big variations are there, like here you can see only D1074 has the sequence and the remaining all has, there is no sequence. That means these are all that nucleotide sequence might have deleted. Okay, this we can use it as a marker, indel marker, either insertions or deletions. So like this, we have to align the sequence, and this variation will be used for construction of a phylogenetic tree. So here also you can see from the ancestral species after division, again they might have attained the mutation, and when you align these two populations, then we will know where exactly that mutation have occurred and what is the effect of that mutation. Okay. So third one is the tree building method. So this is actually a very big topic, tree building method. So I will just simplify it. There are two types of methods we can use. These are two types of data for construction of a phylogenetic tree. One is a character-based method. Second one, distance-based method. Character-based method means, so as I was telling, we have a species like A, B, C, D, 5 species we have a sequence. We just align it, you will get the variation. This variation can be used directly for construction of phylogenetic sequence. So here we will use the Mega software. Okay, and for distance-based method, instead of using the character, we have to transform those sequence data into distances like similarities or dissimilarities, and this matrix can be used for construction of a tree building. So basically, we can classify all the methods into these two types. So these are all different phylogeny reconstruction methods: maximum parsimony method and neighbor joining, UPGMA, least square, minimum evolution, maximum. And I don't want to go into the details, but many publications, they ask either neighbor joining method or UPGMA method for the publication purpose. So I will demonstrate only these methods. So these are the simple explanation for this maximum parsimony. Parsimony, nothing but simplest explanation. Okay. So for example, here 4 fishes are there. There are different rights are there. With 5 steps, we can group these fishes, or with 4 steps also we can group these fishes. But in the maximum parsimony, it prefers only which takes less number of steps to construct a phylogeny. Okay, this is maximum parsimony. This is simplest explanation. So in case of the distance based method, as I told, UPGMA, that means unweighted paired group method using arithmetic averages, and neighbor joining clustering method. These are the 2 methods we will be using. So I'll just explain what is UPGMA. So here it actually assumes the rate of evolution is constant for each species. So for example, here 5 species are there. So based on the available data, distance data, A and B are closely related, and C and E are closely related. Then after forming the closely related data, what is the next closely related? Like here you can see C, E are distance is very less, so they are closely related. So to this E and E, which one is the closest? So next D is the closely related. Then you take it as a, it will take it as a one. Then like this, it will try to, it will form a one groups using this method. But in case of the WPGMA, it gives different weights to the distances. So these things you don't worry about this. So the other one, neighbor joining method. This is also one of the most popular distance methods. Why? Because everybody uses neighbor joining. It is a very quick. Okay, with less time, we can find the best tree. With that too, with a large data set. Nowadays, we want to do some 100 samples, 200 samples. We want to classify. Okay, so in that case, we have to use this neighbor joining method. Suppose in the neighbor joining method, let us assume A, B, C, D, E, F all are equally distant. Initially, we hypothesize, but based on the data, first A and C are separately they are close, and C and D are close, and E and F are close. So like this also will give calculate like UPGMA one. So here you can see first the combination of A and B, and combination after AB, then it's the AB with other, and after ABC with other species. So like this, it will calculate all the distances. So other this least square method, minimum evolution method, they are not much different compared to others. But least square method is they consider best distance method, and the minimum evolution is also similar to maximum parsimony. And the maximum likelihood method is, this is the most precise and intensely used method, but it cannot be used for large data sets because it's a very precise. Only with few samples we can do this maximum likelihood. So overall, here you can see if you have a few sequences or few samples, means less than 10, better to go maximum likelihood, because it's always the best method and which gives most precise phylogenetic fit. But if you have a 10 to 100, You can use other methods, maximum likelihood, maximum parsimony. If you have a large number of sequences, better to go for neighbor joining or UPGN because this is very fast neighbor joining. That's why many people use just neighbor joining only, because with the 10 samples and 50 samples, you cannot publish a paper. So you increase the number of samples, naturally you have to go for neighbor joining method. Okay. So as I was telling, distance-based methods are always fast. Maximum parsimony, when you have a strong sequence similarity, you can go for it. And this maximum likelihood is very slow. And most packages uses almost all these methods. Coming to the final step, that is assessing tree reliability. Now we have constructed the tree. Then we have to assess it. How confident are we about this phylogeny, whether it is a right tree or not? How do you assess the tree? There are three methods are there: bootstrap method, jackknife method, and Markov chain Monte Carlo method. Okay. So coming to the first one, bootstrap. So here you can see which evaluates the internal branches. Suppose this is our data. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is actually our samples and this is the sequence. Now what it software does, every time it shuffles the sequence. Like instead of 1, 2, 3, 4, 5, it tries 2, 5, 7, 9, 2, 6 like this, and 5, 2, 7, 1, 9 like this. So every time it changes the order of the sequences and it will construct a phylogeny. Okay. So like this, it will assess whether the tree is how many times it is coming correct when you use the normal sequences like this. Okay, this is the bootstrap. So jackknife also same thing. Okay, so the jackknife, what happened in the bootstrap, it will replace the one sequence with other. It will not delete. Like here you can see 1 is there, 2 or 10 are there. Here also 10 will be there. Only thing is it is replacing with one sequence with other. But in case of the jackknife, it will remove that sequence and see the result, phylogeny tree. Okay, that is a major difference. So these are the some of the popularly used softwares are there. We will be demonstrating now. So is it okay? Or any clarifications required up to now? This is the theory part. Now we are going to practical part. So is my screen visible?
Yes, sir.
Okay.
Yes, sir, it is visible.
Now, as we have promised, today we will be demonstrating three softwares, that is DARWIN, Mega, and Structure. Okay. So from practical point of view, I would like to explain these things. Okay. So suppose we have taken here rice seeds. Okay. So let us say we have some 100 rice samples are there. Okay. We want to know the how they are related. Means whether they are making any group, or like here you can see morphologically they are different. Right here you can see some are orange type, some are black type, some are with arms, some are without arms, some are slender, some are bold type. Suppose if you take the phenotype or you isolate DNA, whether this black type are coming together or not. Okay, or varieties with the arms are coming together or not. Like this, we can assess the thing. So what we do, first we go for, we have to evaluate the samples in the field condition. Like suppose 100 samples are there, we grow the all 100 samples with using RBD or augmented block design or whatever method you apply. You collect the leaf samples, you extract the DNA, you isolate the PCR, means you amplify the DNA with the PCR. You can use the markers most preferably which covers all chromosomes of the organism. If you take more markers, that will be always good, and their distribution should be uniform. They should be distributed on all the chromosomes. Okay, and after amplification, we run a gel electrophoresis. Then we have to go for marker analysis. Means we have to run the amplified samples in the agarose or polyacrylamide gel electrophoresis. Okay, so up to here we do usually, right? Then so from here we have to start. So now we have a agarose gel picture. Now we have to do scoring. Okay, so let us assume this is the dominant marker like RAPD, AX or ASLP. These are variety means different varieties. These are the primers 1800, 1700 and all this. Okay, so these alleles, or we can say bands, this should be converted to scoring, right? So what you have to do, whenever you take first one variety with one primer, suppose one band is there, you will give one score. In the variety 2, the same primer, there is no band, then you give 0. Variety 3, variety 4, 5, 6, 7, 8, 9, 10, all bands are there. Then you give 1, 1, 1, 1, like this. And the same thing for 1700. 1, 2, 3, 4 bands are there. 5th, no band. 6th to 8th bands are there, but 9, I think it should be 0. Okay, so like this we have to score. Okay, so this is for RAPDs or ISR, or you can say dominant marker. So same thing we have to follow for codominant markers, or you can say simple sequence repeat marker. So usually what happens in a simple sequence repeat marker, we expect only one band or two bands, or right? See, this variety 1, when we amplified, we got band at let us say this is a 200 allele. Okay, but variety 2, so here 250 to 200, 150 and 100, 4 alleles are there. Okay, in the variety 1, at 250 no band, so you keep it 0. At 200, yes, band is there, you keep it 1. At 150 no band, 100 no band, keep it 0, 0. In the variety 2, like this, only 100 only band is there, will keep 1. So like this, for all the varieties we have to score. Is it clear for all? Because this is crucial, because this is the beginning point, so everybody will get confused. Is it clear?
Sir, what about
Sir,
codominant markers?
one question.
Yeah, tell me.
SSR or codominant, how do we find it?
How do you find it means? This is the agarose gel picture. When you load in
How
the
do we
agarose,
find that both the alleles are having it or single allele is having?
see, in the agarose you will get only one allele. Like suppose when you load in the agarose gel electrophoresis, when you load in the 50 base pair ladder, you see the band is matching with 200 base pairs or 150 base pairs or 100 base pairs. Okay, or why it is coming full screen only? Okay, I'll do one thing. Now, can you able to see my screen?
Yes, sir, it is visible.
Okay, so you just keep one line here. Okay, let us assume this is a 250 base pair allele. Okay, here you keep 0, 0, 0 because there is no band. Only keep 1 when in the variety 7. In all other 0, 0. Is it clear? Like this, you go to the second allele. Band is there 1, 0, 1, 0, 0, 1, 0, 1, 1, 0. Right? So like this, you have to score all the varieties with all the alleles. Is it clear?
Excuse me, sir.
Tell me.
Sir, sometime marker give a more than 2 band. Then how we can interpret that? How we can generate the data?
Yes.
0 or 1?
Good question. Because sometimes what happens, this is the simplest explanation I am giving. But you take some polyploids or sometimes heterozygotes will come. In that case, what will be there? 2 bands will be there. It doesn't matter. If band is there, you keep 1. Otherwise, keep it 0. How does it matter?
And if
Okay.
there is a 3 band, then?
Okay, you keep it. Whenever band is there, you just keep it 1.
Okay. Thank you.
Okay.
Okay.
So, we have scored the allele using the RFPD or dominant or codominant marker. Okay. Then this is just for your understanding only. Software anyway we will use. Just how software will draw a phylogeny tree, I am going to explain here. So here, let us say this is a construction of a phylogeny tree using the character matrix. So this is the operational taxonomic unit. Let us say, or we can say 4 varieties, 4 species.
Sir, please keep it in presentation mode, sir.
Because 1 second. Okay, right. Okay, so here these are the 4 species are there and 10 markers are there. Okay, we have scored based on the agarogel picture, we have scored 0, 1, 0, 1, 0. Now, how the software will convert into a matrix? Let us see the first one, and we have to make a checkerboard like this: A, B, C, D, A, B, C, D. So first you compare A and B. Okay, how many are matching? Means we are calculating the similarity matrix. So 0, 0 matching, yes. Here 1, 1 matching, 1, 1 matching. Means first sample, 8, 9th sample are matching out of 10. That means that is 30%, right? Then you see the A and C. You see the commonality: 1, 0, 0 matching, 1, 1 matching, 2, 0, 0 matching, 3, 0, 0 matching, 4. Only 4. That means 40%. Like this, you see A and D, 70%. 1, 2, 3, 4, 5, 6, 7. Out of 10, 7 alleles are matching, so it gives us 70%. So like this, all probable combinations, we have to calculate the similarity matrix. Is it clear?
Yes, sir.
Okay, very good. So now using this similarity matrix, how
Sir, in case of SSRs, sir, it will be for one primary, right, sir? This case.
No, even SSRs also, we are pooling the all data. See here, this I have showed only one allele. I told you to use the markers which are distributed on all the chromosomes, like 40 markers, 50 markers. You score all the markers, you pool the data. and give it to the software, right? We will not calculate for each marker. Is it clear?
So, sir, basically, sir, in this case, the alleles, we have shown 4 alleles of one single marker.
Yes.
Whereas in case of ISR, it was the different case.
Yes. But whatever may be the case, you have to pool all the data and calculate the similarity matrix. Okay. So now, based on this checkerboard, now tell me which species are closely related.
A and D, sir.
A and D, sir.
A and D, very good. Next.
B and D. Sorry, B and C, sir.
Yes.
B and D.
So out of 4 species.
B and C.
Okay. So out of 4 species, based on the similarity matrix, you can say A and D are similarity matrix is high, so they are forming a one group. And B and C similarity matrix is high, they are forming another group. Okay. Now we know which species are closely related. Now what about their among relation AB to CD? Okay. For that, what we have to do, you have to take the remaining similarity matrices. Like other than these two, 50 and 70, you just leave it. Remaining, take it. 30 plus 40 plus 40 plus 30 means A to B and other things, right? Then you make it 140 divided by 4 will be 35. So 35 actually root from where the common ancestor has come. Okay, and from 35 onwards, these 4 are different. And A and D, what is the similarity? 70%. You see from left to right, at 70%, they have a similarity matrix. And B and C, they have 50% similarity matrix. Okay, so like this, the software will draw the phylogeny tree. Is it clear for all?
Yes, sir.
Yes, sir.
Okay, this is just the simplest explanation, but the practically you will see a lot of difficulties. Okay, this is just for your understanding. Now I ask you, I request you to open the software. If you have already downloaded, then it's fine. If anyone not downloaded, you just follow this instruction. And this PPT also I already shared you, right? So you just type Darwin, the Google. So the darwin.carad.fr will come. You just double click it. You will get this window. So here you fill the just registration, your email, name, organization, country, and everything, and it will immediately start it. Means it will immediately download. After downloading, you double click that software. Their application part will come. Now always softwares are raw files will be in application form. So you double click this Darwin 6. You will get this window. Are you practicing now?
Yes, sir.
Because I already provided you the data files also. So simultaneously we have to do. Don't think this is a theory and practical will be separate. Okay, simultaneously practice it. I will ask you to show the results after this. I will give the permission for screen sharing. You have to show your results. I'll pick up randomly. I'll ask you. Right? So here, first thing you see under the dissimilarity, there are 3 files can be used. One is the single data, and second one is the allelic data, and third one sequence data. Single data is nothing but this one, 0-1 matrix. That means this is score of dominant markers, mostly RAPDs or SSRs, ISSRs or AFLP or something like that. Okay. Then this is the single data, and the sequence data. Sequence data is nothing but SNP data. If you know AGCTs, which variety has A, which variety has B. So this also we can use it. And the third one is the allelic data. Allelic data here you can see this is the variety left side. The top one is 2 times we have to give because by default we should consider it as a diploid. Suppose if I give here a rice, so rice is a diploid. So each time you have to give 2 alleles. Suppose if you are getting the same band, you have to give the same allele size. Okay, 105, 105, 105 like. Suppose if you are getting any heterozygote, means one allele 105, another allele 130 or so. Here, yeah, here you can see 266. One allele has 110, another allele has 130. So in that case, you have to give different allele size. Okay, so in that way, 3 files we can use it. I think all two files I sent you. Okay. So you just first we will practice with the single data, that is a 0-1 matrix. So here what I will do. Okay, I cannot minimize it. Oh, there is a problem. So you have this file. You know, this Darwin, they have a certain format. It starts with the @ and Darwin 5.0 single should be there. Here number of accessions means number of varieties or species or whatever is there. Okay, and here let us say for the demonstration purpose 3 markers you can say. Okay, these are the code ones. Okay, then at the end if any missing data is there, you have to give 99. Okay, so here we have to give some name. After that, you go to the file, save as. All are seeing my screen, right?
Yes, sir.
Okay, here you go to the text tab delimited. Okay. After that, you give var. That's the file format which accepts by Darwin. So this is a file, right? Then go to the dissimilarity, press the calculate from single data. So this is the workshop single var file. You open it. Suppose if any missing data is there, you have to click this. Some data are missing, and what code you have given. Usually we have to give 99 or something like that. Whatever you have given, you have to give that.
Hello, sir.
Yeah, tell me.
So screen is
Sir,
not
screen is not changing, sir.
What is the problem here? Okay, what about you? What about now?
now it is okay, sir.
Okay.
Yes, sir.
Okay. We press. I will close this one. We go to the dissimilarity. Press calculate from single data. You press this workshop single. So this we have converted into WAR file, right? Press open. Then if any data is missing, data is there, you press click, some data is missing, and we give 99 code. Okay. And number of bootstraps. Usually for publication purpose, they ask as many bootstrap values as possible. Usually 1000, 2000 is okay. Okay. For the demonstration purpose, I just keep 100. Okay. And this other options, some people use dice and some people use Jacquard. So I think anything can be used. Then press the save dissimilarity. Okay. So in the same folder, it will save as a .dis file, means dissimilarity file. Just save it. Okay. So now it has calculated. So everybody completed that one?
Excuse me, sir.
Yeah.
I could not upload the file to this Darwin software.
What is the problem?
The file format is not showing when the open window appears.
No, have you done this one?
Yeah, I changed the Excel file to
dot file. Did you convert into .war
yeah
file?
Yeah, I did to the text delimited something.
Is it showing war file here?
No.
Then you do one thing.
Maybe it was not mentioned .var, sir, there.
Yeah, you have to mention it. See, as I told you, you go to the file,
Okay.
save as, here, save as type.
We cannot see. Yeah, okay now.
Text tab delimited. Just put .var.
Okay. Okay.
Okay.
Okay, sir, thank you.
What about others? Is it okay?
Yes, sir. Yes, sir. We are following.
Okay. So dissimilarity, we got this file, right? Everyone got this file, .this file. So if you want to see the bar graph.
Sir, I'm facing the same problem as for the earlier person. I have saved the file in text delimited and the var as well. But when I open the Darwin software, the particular file is not being shown, sir.
I don't know your system configuration. You have to see that. What about other?
They have to opt for the all files option, sir. Then it will appear.
No, it should show as a
.var.
.var. Then only it will recognize.
Yes, sir.
No one got. This file. Hello.
So kindly type the extension name .var.
Or you can do one thing also. Okay, you can go to, can you see my screen? Hello.
Yes, sir.
Okay, so you can go to the software where your software has been downloaded. Go to the data file. Okay, so here also war file will be there. Okay, you do one thing, you just remove that and you paste your file. Okay, and just save it. Like this also you can try. Did you understand? If any, I think I already mailed you some var and don file. Okay. That also you just replace the existing data with this data. Simply save it. Any problem? All are facing the same problem?
Yes, sir. Yes, sir. Yes,
Did
sir.
you, did you try this one? No, did you try this one? This is a example single file, right? Okay. You just
Sir, can see your screen, sir. It's not moving, sir. It's stagnant.
my screen is not visible.
No, sir.
Yes, not visible, sir.
What about now?
No,
Yes.
okay, sir.
Now it's visible.
Yes, sir. It's visible.
So here the Darwin example files will be there. The Darwin software when you download it. Okay. Here also 3 files will be there: single, allelic, and sequence. So now we are testing single. You remove this data, you paste your data in the Excel file. Just save it, and no need to save again var file. Still not coming.
No, sir.
In that case, we can do already existing data, because in my system it is coming. So simply I am doing like this only. I am not doing anything. Or shall we use already existing data? Hello.
In program files, no data is there, sir.
Yes, sir.
No
It's a
data.
blank. Yeah.
Yes.
Yes, sir.
No, in
Showing
the
blank.
no. See, this file should be there once you download the software.
Sir, they have just provided the setup file, sir, not the complete folder. I think.
Can you able to see my screen now?
No, sir.
Visible, sir.
Now?
Yes.
Yes, sir. Visible, sir.
This data folder is not there.
Data folder is there, but it is empty.
Empty means this thing should be there. I think some problem with your Windows. All are showing like this only.
I don't even have this, sir.
How come?
I just have the setup file.
No, all these things should come. All these DARWIN.
We got all these files, sir, but in data, nothing is there.
So in my data, it is there.
What about others?
It is there, sir. It is there.
Yeah, I think it should be there because I didn't do anything. With the software only, they provide this sample file. Anyway, I sent you morning one war file and dawn file. You can try with that. Shall we try with that?
Yeah, okay, sir.
So this is the
War file is not opening, sir.
no. You go to the dissimilarity, single data. Did you get this GS5 and PhD file?
Yes, sir. GS5 and PhD I got, sir.
Okay, you press one war file, GS5. Okay, and you set some data is missing. Keep it 99.
Okay, sir.
Okay, and keep it bootstrap value 100. And save dissimilarity GS5 only. No need to change. Just save it and click OK. Up to here completed. Completed.
Yes, sir.
Okay, then you go to the factorial analysis. Okay, yeah, analysis. You click the gs5.disk. Okay, press open. Then save factorial coordinate, and it will save as a .vft, the same folder. Okay, save it. And you click this display when done. Then no need to change anything. Press OK. Did you get this file? Factorial analysis.
Please, can you repeat once again, sir, from after the disk file creation?
See, after the disk file, you go to the factorial analysis. Okay. Can you see my screen?
Yes, sir.
Okay. You press analysis button, select that disk file, open, then save factorial coordinates as. So in the same folder, it will save as a .aft. Okay. Because we already created, just replace it. You press this display when done. So when the analysis complete, it will show
Excuse me,
the.
sir.
Yeah, tell me.
I think I could not save the dissimilarity file.
Why?
It's not showing again. I saved it, but I could not get it again. For
Yes, sir, same problem.
analysis.
I don't know. I think some voice problem. All are getting same problem.
No, sir.
No, sir, I'm able to do this now.
You can able to do.
So actually, the first file which you had said as text delimited, it should be no. I think the normal text file format, sir.
Okay.
I did that, and now it's being shown, sir.
Okay. So once it is done, you press OK. So it will show the factorial analysis. Everyone got this factorial analysis figure?
I got it, sir.
Okay.
I got it, sir.
Okay, good. Okay. After that, you go to the tree construction. You can choose mostly neighbor joining. Let us use it. You click the same disk file, open. Okay. And here we use UPGMA, unweighted neighbor joining. Okay. And bootstrap values, click 100 and click draw tree when done. Right. And you can select the identifier file also, that's DON file. And you save the tree as .arb file, it will save and press OK. Okay, so like this you will get the dendrogram. Anyone got it?
Yes, sir.
Okay, so here there are so many options are there, hierarchical and horizontal, unrooted, growing from root. So many options are there. Okay, so here if you want to show the bootstrap values, you can show the bootstrap values. Scale also it shows, and identifier file if you select it. So we can give the names also for each variety. Okay. One second.
Hello, sir. Hello.
Tell me.
Sir, how to change the number to accession from identifier file?
So here, here it is there, na? Open identifier
Yes.
file. You click this
Okay.
accession.
Okay, okay.
If you keep it
Open
unique,
identifier. We need to load the any file or directly it will come?
no, actually, we have to create a DON file. I showed you, no?
Yeah, yeah. DON file.
This 1 second, I'll show this. So here we have to click the identifier file.
Okay.
When you are doing the tree construction, you click the DON
Yes.
file. You click bootstrap analysis, draw tree when done. and save
Okay.
tree as remaining all same. You keep it. Okay, after that it will show the result. Again, you have to select here identifier file. Then you press the, instead of unit, you press the accession.
Okay.
You will get the correct image.
Don file is must for changing
Yes.
number to accession.
Yes,
Okay,
yes.
okay, fine.
And another beauty of this Darwin is we can give colors also. Let us assume I know this 4 are from irrigated ecology. I will give one color. Suppose if another 4 or another 5 are of different types, I can give different color. So here in the diagram. Okay. So in that way, it will give very beautiful picture. Of course, here based on the data, it has given like this. With PhD file, can anyone try and show me? I will give the permission for a share screen quickly.
Sir, how to colorize the group means according to groupism, how we can colorize?
So here left side one is there, madam. Open unit color section window. Can you able to see this 25? You click this one.
Okay.
Now you can give the color.
So it don't give the means directly. We have to provide the link, means we have to provide that one in.
We have to provide the color. We have to give. You select your varieties, and you can give any color.
Okay, sir. Okay.
Sir, I could not get the Dawn file, sir. From its created, sir. I could get this var as well as a single file, the this file, but Dawn file I couldn't get, sir.
I sent you by mail. Or with another file is there, PhD. That also you can try.
Sorry, sir.
Okay, sir.
Excuse me, sir.
But basically, I'm using the Excel file which you are saying, sir. Workshop single. I'm using that file. For that also, we can use the same Dawn file, sir.
For that, you have to create a Dawn file, because the software means that file you are using, that single file.
Yes, sir.
In that case, you go to the file, import data matrix. Okay.
Okay.
This is the file you are using, right? Workshop
Yes,
allele.
sir. This
is the file.
I'm using
right?
a single one, sir.
Okay. So if you click this one, you can open it. You can click allelic. Don't change, because ploidy is only 2. Save data as in the same folder. It will save it as a var as well as Dawn file. 2 files might have come now. Did you try this one?
Sorry, sir.
Yeah, tell me.
I'm quite behind. I got the disk file now. So how do I proceed from here? Can you please show it quickly?
Actually, I provided the PPT. So this file you got it, right?
Yeah, yeah.
Suppose this factorial analysis have you done?
Yes, yes. Now I am here. Yeah.
Okay.
Despite no. I am in the analysis. Yes.
Okay. You click the factorial analysis.
Yeah.
Press analysis. You select that disk file.
Okay.
Whatever is there. Okay. And you save coordinates. In the same, it will save it as a .aft. You click this display when done. Press OK. Now it will show the factorial analysis.
Yes, sir. I got it.
Okay.
Okay.
Then you close this. Then you go to the tree construction. Go to neighbor joining. Press again this relic or whatever disk file is there.
Okay.
Then you select the UPGMA, unweighted neighbor joining.
Okay, sir.
Press the identifier file and draw tree when done.
Okay.
Save tree as. then press OK.
Okay, sir, got it. Thank you.
Is it clear for all? If it is okay, then we can move to the next software, Mega. After a 10 minutes break.
Sir,
Tell me, madam.
excuse me, sir.
Yeah, tell me.
Sir, I have a doubt. Sir, I have a doubt. The maximum number of missing data that can be used in this DARWIN software, sir?
Actually, I'll show you, madam, here in the beginning file. When you press this dissimilarity, let us say allelic data which you have taken. Here we have to press, if any missing data is there, we have to press this one. Okay. So here different proportions are there. I am keeping the missing data, minimum proportion of valid data required for each unit pair is 90%. That means if any data or any species having the 90% below data, it will not consider the software.
Oh, okay.
It will consider only above 90%. Suppose if you are not confident of your data, you got more missing values, then you can reduce yourself 70%, 80%. Okay, like this you can do it.
Okay.
And whatever code you have given, you can give the same thing here. Right?
Oh, okay, sir. Thank you, sir.
Anything else, or shall we move to the next session?
Yes, sir, we can move.
Shall we have a break, or after Mega we'll have a break?
Five minutes break and then continue, sir.
Tree, what that numbers indicate 3377, Bhavya. So they are actually bootstrap values, Bhavya. So bootstrap values above 70% is considered as a good. That means that association is not random. That association has some meaning. That means they are associated evolutionarily, not by chance. Okay.
Thank you, sir.
Okay. After 10 minutes break, shall we start Mega? Meanwhile, with you can fix your problems that this file. Okay. We will back after 10 minutes. I'm back. Are you people ready?
Yes, sir.
Yes.
Yes, sir.
So I request you to see the PPTs which I already sent you. Those who are in advanced stage, they can go to the Mega directly, because in the PPTs, everything is self-explanatory. If you have downloaded the software, the example files I have given, you can directly follow the steps in the PPT. Okay, anyway, I will show here demonstration. So everybody downloaded the Mega?
Yes, sir.
So let me see anything we missed. I think we just follow these steps. Okay. And somebody was asking how to interpret the results. Can you able to see my screen?
Yes, sir.
Okay. So this is a phylogram, we get it. Okay. So here you can see these 24, 21, these are all our variety names or accession names. Okay. And these 89, 75, these are all bootstrap values. You know, always bootstrap values above 70 is considered as a authentic relationship. Like you see, this is 100%. The relationship is 100%. Means it's a genuine. Suppose you see this any relation, like here it's showing only 29% or 24%. That means that relation is not evolutionarily conserved. It may be due to by chance also, right? So here we can see from the root, if you see overall, this dendrogram has classified into two parts. This is the one part, this is the second part, right? Within a one part, again we can see 24, 21, 25, 23, these forming a one group, right? Like this here you can see 20, 19, 29, like this, they are forming a one group. So like this we can discuss, depends on your objectives. Suppose we have used different drought tolerant lines with susceptible lines. Are they making separate grouping, like all drought tolerant lines are making one group and susceptible lines are making another group? So these things we can see using this Darwin software. Okay, so this is the interpretation. So this is the assignment you can do using those files, PhD file or GS5 file or something, right?
Sir, just I have a single doubt, sir. So
Yeah.
after the construction of the tree, sir, at the both the ends, at the root section as well as the towards the node section, terminal nodes, I find a line of halo-like thing. What is that actually, sir?
What is that line of?
A line of values or something like black dots, sir, at the terminal as well as the root part, sir.
Terminal is a terminal node, like a variety or a species zone.
Sir, but they come in a huge numbers, like the black dots, a number of things like that.
So many have come.
Yes, sir. Not a single ones.
Can you share your screen? If you have.
Yes, sir. So this is the thing, sir. I get like this, sir. Huge number of dots, sir.
Acha, this is the file which I given, right?
Yes, sir. Yes, sir.
That means there is no difference between these varieties. Means all are forming a same group. That means there is no sequence variation or there is no marker difference. Okay.
Okay. Okay, sir. Okay, sir. Thank you, sir.
Okay. Right. So shall we move to the other one, Mega?
Okay, sir.
Okay. Can you all see my screen?
Yes, sir, visible.
Yes, sir.
Okay. This is one of the popularly used software. Mega means nothing but molecular evolutionary genetics analysis. It has a 35,000 citations, right? So based on this, you can imagine the powerfulness of this software, and this is freely available and user friendly also. And one more important thing is it is developed by one Indian called Sudhir Kumar. Of course, he is in US only. He is in Temple University, but he has a very reputed evolutionary biologist. So he only developed this one. So we should be happy for that. Okay. So directly we'll go to the practical. So I hope many people all are downloaded Mega. If you're not downloaded, you
Yes,
just
Yes,
type
sir.
Mega software. You will get all these things. You can press click, and here you can simply download it within few minutes. You can download it. So this Mega 64 application part, if you double click it, it will open this diagram major thing. Okay. Then first you go to this first one, that is the alignment or edit or build alignment. Okay. Maybe here you can see whenever click this one, if you don't have a already aligned one, you are creating a new alignment, you just press create a new alignment, press OK. So now I think I might have given the DNA only as a example file. You press DNA, then you will get this alignment explorer. Okay. So I think better to practice rather than telling. Is it clear? My screen is visible?
Yes, sir, visible, sir.
So you got the file, alignment file, you got it? I think in your mail you might have seen. 1 second. This is also fine. This, did you get this one? DPH123 notepad.
Yes, sir.
This is also fine. Our cluster file. This is also fine. Right? Did you get this cluster file?
Yes, sir. Cluster
Yes.
file is also there.
Okay, good. So in that case, this Mega is very simple. You just paste it, copy paste, Control C. Okay. You go to this alignment, edit build, create new alignment, press OK because it's a DNA. Press OK. Here, just right click it on sequence, paste it. Did you get this one?
Yes, sir.
Okay. Now we have retrieved the data. Now we have to align. This is just a sequence we have imported. We haven't aligned. Okay. So under the alignment, there are two options are there. One is called cluster W and another one is the muzzle. Okay. So here codons means whenever you have a cDNA sequence, in that case you can press the codon because this is a normal DNA. You just either press cluster W or muzzle. And out of these two, muzzle is most popularly used and it is very fast also. Okay, so we will follow this one, align by muzzle. Press this one. Okay, we will run with the default options. Okay, so no need to change everything. So press OK. Now it has completed alignment. Did you complete the alignment?
Yes, sir.
Yes, sir.
Okay. So in the alignment file, if you see, some people might have got, suppose in some cases alignment is not proper, or it may not give some information. In that case, we can simply delete it. You just highlight this, you can press this cut button. It will remove. Okay, if you don't want any alignment in your file. Okay, then you go to the data, export alignment as mega format. Okay, if you press that one, you can give what is the file name? Let us say workshop. We'll save it as a like workshop 1. I'll save it. So it will save as a mega file, workshop 1. And here also you can give, or you can give rice. Okay, this is a protein coding nucleotide sequence data. Yes. Okay, we completed. Then you close this window. Everyone saved it into mega file.
Yes, sir.
Yes, sir.
Then you go to the phylogeny. So here many options are there. Maximum likelihood, neighbor joining, and minimum evaluation, and so many things. So we will go for this neighbor joining. Okay. Then you select this neg file, whatever name we have given, workshop 1. Okay. So here under the analysis preferences, test of phylogeny, we should select bootstrap method, because this is very, very important. If you want to publish in a good journal, you have to follow this. You have to test this phylogeny. And the number of bootstraps also minimum 1000. Okay. So for the sake of demonstration, I kept 500. So the remaining no need to change. Substitution type and everything, let it be default options. Simply press OK. Completed. What is the problem? Did you get phylogeny? Or you also got same problem?
Yes, sir. We didn't got, sir.
Okay, let me see. I think we have deleted something, so that's why it is showing some problem. Paste it. Okay, we paste it. Go to this alignment, muzzle, align DNA.
Sir, that seventh sequence need to delete, sir, or that one, sir?
Oh, okay. How come it has come? 7th one?
Yeah, that one we need to. Yes.
Oh, okay. Okay, now we have deleted. Now we will save export session in mega format. We'll give same file name. Price. Okay. Now we will save it. Some noise is coming. Okay. Then we'll go to the phylogeny, neighbor joining, bootstrap. Okay. So again, problem is coming. So again, it is showing the 7th sequence. How come it is showing the 7th sequence?
Sir, old it is saved, I think. We need to delete that processing file.
How come here there is no 7th?
So now it's showing the phylogenetic tree.
It's showing. Very good.
Yes, sir.
Let me see. Yeah, now it will show. Okay, now it has come. So that 7th row is the problem. Everyone got this?
Yes, sir.
Yes, sir.
So here also you can see these are the similarity indexes. This is the bootstrap values 93, 99, 63 and all these things. Okay, and here you can see some more options are there. This is the traditional file. If you want, you can convert into a straight cladogram, or if you want to convert into a curve, or you can convert into a circle. You can do many things, right?
Sir,
Yeah, tell me.
sir, what is the difference in cluster W and muscle?
Now, cluster W is a little bit slow, and muzzle is very fast. That's what I understood. But actually, algorithm wise, I don't know how they are fast and how they are slow. Okay.
Which one is better? For,
Yeah, muzzle is the better one.
Oh.
That's why we are doing this.
Okay.
So you can keep tinkering with these options. So you can find many things. No, you can swap the subtrees also sometimes. So here you can see bootstrap consensus tree. Everyone got this? Hello?
Yes, sir. Yes, sir.
Okay. Now, so that is a straightforward method of using this one. Okay. So I think one file I have given you as an assignment. As a, what is the file name? Workshop. Huh? In this one only, workshop. BPH 123. So this one you can try. Try and show me anyone. I'll stop my screen. I'll pick up randomly. I'll call by name. So whoever completes, they can show the results, dendrogram.
Sir, just as you said, whenever we select the files and align, so if we delete that non-aligned file, that is the dots, if we delete, will there be any problem, sir, in creation of the tree, sir? Hello?
I don't think it is a problem. I think
Okay,
if it has some insufficient data, so better to remove those kind of sequences.
Okay, sir.
I got it, sir. I got
Okay.
it.
You show it.
Mm,
Share your screen.
okay, sir.
Oh, very good.
Can you see my screen, sir?
Yeah. Okay, very good. Good. What about others?
Yeah, done, sir.
Okay. Shall we move to the next one? Final structure.
Yes, sir.
Yes, sir.
And here one more thing is there. If you could close the screen, I will show you. Prajwal, huh? So you can save those pictures. Can you all see my screen?
Yes, sir.
Yes, sir. Visible, sir.
So here you can see the bootstrap values. So it should be always more than 90%. Then it is strongly supported values. So 70 to 90 also okay, but 50 to 70 is very weakly supported, and below 50% is hopeless. That means we cannot conclude anything if bootstrap value is below 50%. Okay. And the other thing is, you might have seen this scale bar that indicates the number of substitutions per scale. Okay, suppose if the scale bar is 0.020, that means 2 base differences for every 100 bases. Suppose if the scale bar is more, that means here you can see E. coli is more distantly related to the Shevanella algae. So like this, we can see the number of substitutions. So this scale bar indicates that one. So for the publication purpose, bootstrap values should be there and the scale bar also should be there. Okay. So this is you already done. Now, can you see my screen, mega screen?
No, sir.
No, sir.
Can you
Okay, now
can you repeat step by step from starting onwards, sir?
Yes, sir.
Yes, sir.
Yes.
Okay. So one thing you have to remember, when you have a molecular marker data or microsatellite marker data or at the max sequence data, you can go for Darwin, right? But nowadays all our sequencing based results are coming. So in that case, we can go for mega. Okay. So if you have a sequences, directly you can paste it and you can draw the diagram. Okay. Now one more option is there. There are so many sequences are available in the database, like NCPA and all those things, right? Suppose we have identified one gene, we have sequenced. We want to know the association of our gene with already existing one. Okay, in that case, how do you use? In that case, we can go for a mega only. Go to alignment. Here we can go for show web browser. So it will directly take you to the NCBI file, NCBI from mega. For a normal running mega, it doesn't require internet. But if you want to run through NCBI, you need an internet. Can you able to see this screen?
Yes, sir.
Yes, sir.
Okay. Now I want to test GA20 oxidase enzyme. I want to test this gene has any orthologous, means the similar kind of genes are there in other crops or other organisms. Okay. Now we have to go search. So it picked up. So it is showing it is there in the rice. Okay, good. Located on chromosome 1. And the same thing it is showing on Solanum tuberosum also, potato also. Okay, no problem. I want to see the how much closely related the gene in rice with the potato. And here also GM is the second one. Okay, and you can click couple of things. Okay, and can you go to okay, in that case, I think we have to add one by one. Okay, just leave it. Okay, press this one gene. So it will take you to the gene sequence. Okay, and summary and everything it will be. So here you go to the gene sequence will be there in the graphical format, FASTA format, and gene bank format. You press this gene bank format. Okay, after pressing this, you directly go up in the mega add to alignment file will be there. Okay, you click this one. So it's asking whether I should import only coding sequence or the full sequence. So for the demo purpose, I will import only coding sequence. So it is showing Varaja sativa and Nippon bar, third some locus name. Okay, other just leave it. So it has added, right? Then again, I want how to go back from here. Okay, I'll go from here. Okay, now I remove this one. I will add this GMH maize also. Same way, I will add, click the gen bank. So after showing the result, I will add to the alignment file. Same, import only CDS. It shows GMH. This is the variety name. This is the locus name. Okay, I have added 2. Okay, then I will add one more thing because 2 it may not take. Okay, tuberosum, potato also we will add. So potato also press gen bank. You add to the alignment file. Tuberosum. Okay, locus. Okay, we added all these 3, right? Now we will see same gene in different crops, rice, maize, and potato. Now this is just I imported the sequence from NCBA directly. There is no copy paste. Then again go to the alignment. This is a let us say DNA file. What is that? New sequence needs to be selected. Okay. Okay, select all. Okay, I have selected everything. I said okay. Now it is aligned. Now here you can see the alignment.
Sir, your screen is stagnant again, sir.
Not showing?
NCBI is showing, sir.
It is showing
on the
NCBI?
top of it.
Yes, sir.
Again, I have come to the. What about now?
Yes, sir. Yes, sir.
Okay.
Yes.
Now after importing, the procedure is same. Again, go to the alignment, align DNA, select all. It's already selected. Completed the alignment. Is it okay? Hello?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. Now we go to the data, export mega format. This is from NCBI. Let us say. Save. What is it? GA20 OH. Yes. Okay. Now you close this one. Close this also. Now you go to the phylogeny. Let us see neighbor joining. Same. Bootstrap 500. Okay. What is this? No, it is. Okay. Yeah, NCBI. Okay. What are this?
Sir,
Yeah.
which version is best of Omega?
This is 11.
Okay.
Okay, at least 4 tags are needed for bootstrap. Okay, in that case, we'll remove bootstrap for the time being. Okay. I'll remove this one because only 3 we have done, right? Okay. Now it has come. So, as per this dendrogram, rice and maize are closely related compared to potato, right? So
Right.
like this also we can do it. Okay. Is it clear?
Yes, sir.
Okay.
Yes, sir.
Then shall we move to the final version?
Yes, sir.
Okay, sir.
So this is also a very simple one. Have all downloaded?
Yes, sir.
Yes,
Can
sir.
Can you see my screen?
Yes, sir.
Yes, sir. Visible, sir.
Okay. This is actually, I will tell you the purpose. You know, as I already told you, if you have a molecular markers, you have to go to DARWIN. If you have a sequence data of NGS, or if you want to retrieve from NCBI, better to go for MEGA. Okay. But in the MEGA and DARWIN, what tree we get, that is based on the whatever information we are given, like molecular marker information or sequence data information. That is based on the entire information, it will show which species are closely related. Okay, but in this structure software, within a organism, how many are of some X population allele and how many are of Y population allele. Okay, let us, I'll take the real time real example. Let us say you take Andhra, okay, it's a majorly Telugu speaking state, but still we can find some Rajasthani people, Marathi people, and so many people, right? So here, if you for the overall we are saying Telugu speaking, but within the Telugu speaking there are so many other state people also there, right? Similarly, a variety means it will not be developed on its own. It is developed over the decades by crossing different parents. And here this structure software shows the how many are of suppose if 4 populations are there, how many are of population 1 type of allele and how many are of population 2 type of allele. Like this we can see in the structure software. Okay, this is the basic funda. Did you get the okay? So those who are not downloaded, you just press this Windows button, download it. Okay, directly we'll go to the software now. Did you open the software structure?
Yes, sir.
I think I have not provided you the full, right? I think I have given you the only raw file, isn't it? So because I want to show how to develop the file also. So this is a simple Excel file. Can you see my screen? Excel screen?
Yes, sir.
Okay. So the left side ones are varieties and the right side ones are markers. Okay. And here also it's a considered as a diploid. Means each variety we have to repeat 2 times. Okay. Suppose here how many are there? 100 are there. That means 50 varieties are there. Each variety repeated 2 times, so that you can add whether it is the same allele or different allele. So in that way we can identify any heterozygotes or something like that also we can include it. So 2 allele. Suppose if any missing values are there, keep -1. Okay. So this also similar way, copy, save as. In the same.
Sir, just a small doubt from my side, sir, in this case. So suppose, sir, if a particular variety is having more than 2 numbers of alleles in the same case, sir, like you have mentioned about 105 here, sir.
Yes, yes.
In this case. So what that exactly signify?
Better to remove that marker. Okay.
Okay.
So because the markers which gives ambiguity, so we cannot because the software cannot address those kind of problems unless until if any specific options are there. It will accept only biallelic data, means for diploids only. For for polyploids, I think some other software might be there. Okay, but majority of crops or humans are all are mostly diploids, right? So you go to the file again, save as text tab delimited. Same way, just give str data. At least now everybody could save it. Fill a simple text file. Have you done? Again, problem?
Done, sir.
Okay, good. So you go to the file, press new project. Okay, project name will type workshop. Okay, can you see my screen? Okay, so select directory. That means we have to go back to the software where that file is located, means that folder, structure folder. You just press the folder name. No need to press any file folder name. It is asking the directory. Then it is asking the choose data file. Then go to the browser. Within the folder, this is a data file, just simple text file. Press select, go to next. Number of individuals 50, number of loci 17, missing value -1. Okay, row of marker name, yes, because we have given the marker names in the file. Individual ID for each individual, yes. Okay, and putative population origin, no, we have not given. That means if you already know they belong to some population, like you see in rice, they belong to indica type or japonica type, you can give that out. But I have not given anything. Okay, just finish it, proceed. So now it loaded the data.
Please, can you repeat it once, sir?
Yeah, go to the file. Okay, I'll close this one. It is not taking. Okay. 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 Now can you see my screen?
Yes, sir.
Okay. Go to the new project. The file new project. You give some name, workshop. Select directory. Directory means where the software has been loaded. Mostly in the C drive it will be there. Program file, structure 2.3.4. Just you select that folder. Okay.
Okay, sir.
Then you select the file name. That whatever file we have converted into text, that is the str data. Select that one. Then press next. Number of individuals in the file is 50. Number of loci 17. For missing value we are given -1. Okay. Row of marker names. So for marker names we are given in the file. And individual ID like the variety names also we have given. Then finish, proceed. It has loaded. Got it? Hello?
Yes, sir.
Okay, good.
Sir, just the step 3, we should tick the row of marker names and also
individual ID.
Okay.
Individual ID, because we are given the marker names and ID also. Okay.
Sir?
Yeah.
Sir, actually, I am using varied populations. Different lines are there from distant, some land races, some interbreeded population like that are there. In that case, I get more than 2 lines. At that condition, what I can do, sir? Can I go with this dominant data 0101 like that?
0101 structure will not take.
01 will not take, sir. Actually, I tried like that, sir. I got finally one result also.
No, in that case, you can do one thing. You can separate each allele as a one marker. Suppose RM2,
like that only I have taken.
Yeah, RM224 has a 4 alleles you got. Okay.
Yes,
you take each allele as a 1 marker. Okay. Same for the
Okay,
same variety you give 105, 105, and 110, 110. Like this you can give.
Yes, sir. Okay.
Yeah. In that
Yes, sir.
case you can do that.
Thank you, sir.
So then we'll go to the project. Parameter set you will set up. So this parameter setup here, length of burning period.
Excuse me, sir.
Yeah.
When I went to save the file, when I did the finish button, I am getting a window called bad format in the data source.
Yes, sir.
Yes, sir.
Same problem.
Same problem.
How? This Excel file, huh?
No, sir. After uploading it to the structure software, after pressing the individual ID for each individual, the final step, when I press finish, I'm getting this error.
You know, sometimes what happened in the variety names, there should not be any gaps. That you have to ensure. Sometimes I think I removed mostly yesterday. If you have find out any gaps or any commas, full stops, nothing should be there. If something is there, it will not take.
Yes, sir. It is saying that data entries are more than expected. That is 18 at line 2.
Did you remove the gaps first of all?
No, sir. I'm going now.
I think which file you got? STR data?
Yes, sir. Same.
I think for this file, see here IC between IC and 25 gap is there, right?
Yes, sir.
Yes, sir.
You remove those gaps. Okay, then it will take automatically.
It is actually saying there is one extra entry in line 2, that is 18
Yeah,
instead
yeah.
of 17.
Yeah.
So we just remove.
And I'll tell one more thing also. Yeah, this data is there, no? See here, this is a variety STR data. You remove these gaps here like this. Can you see my screen? Hello?
Yeah.
So, and here allele sizes, sometimes what happens if the name is less, it will come here. So in that case, make it uniform. That is also a problem.
Okay, sir.
So, and here a lot of gaps are there. You remove those gaps. You make it, there is no points also. Here Basmati 370.1 is there. You remove that one also. It will not take. And same name also should not be there. In that case, you give separate name. 371 you can give for the second one. Now, bioinformatics input file is a very important. Once input file is there, then it's easy to run. Like this, you can remove all the data gaps. You know, in order to save time, you can do one thing. You just okay, you just replace this with V1 and V2. Anyway, variety names will not matter here. So you can save time. Are you following? You can replace the names, variety names with some name V1, V2, something like that. Now you can try. Could you fix it?
Trying, sir. Just a minute.
Yeah. Just to save the time, you can remove some accessions or some markers also, so that we can save time.
Yes, sir. Fixed. Thank you.
Fixed. What about others? Those who solved the problem, they can follow that PPT so that you can move to the next step. No need to wait for me. If others are fine, then I will move to the next step. We will move to the next step. Hello.
Sir, I'm facing the same issues. Sir, I have just edited the things, but still the same.
I told you, na, you just remove the variety names. You just keep V1, V2, V3 like this. If any gaps are there, it will not take gaps, comma, full stop.
Sir?
Yeah,
Yes,
tell
sir.
me,
Done,
Girija.
sir.
Sir, why the same variety repeated twice in input data file?
Because it's a diploid, no? Two alleles we are giving. Same allele two times, diploid.
So to indicate diploid nature, we have to repeat the same.
Yes, yes.
Okay.
What, Amit? You are done?
Yes, sir.
Okay. Shall we move to the next step?
Sir, just for the clarification of mine, sir, suppose a particular line, suppose the marker RM224, I have found the particular band in case of 105, that's the base pair length. So what if I have found, suppose 200 base pair? So will that be continued in the same pattern it is been done here now?
Yeah, of course. Sir. Whatever allele size comes, you have to give there. But
Okay,
it's
sir.
heterozygote.
That means the 105, 120, it indicates, sir, basically the particular band size, sir.
Yeah, that means it's a heterozygote.
Okay, okay,
yeah,
okay, sir.
Excuse me, sir.
Yeah, tell me.
For 50 individuals,
yeah,
2 individuals for same markers, we need to upload it for diploid as a diploid.
Yes, yes, yes.
Give it variety 1, variety 2, the data will be wrong for giving in the last file, no?
What? I didn't understand.
If variety 1 and variety 2, I'll just change the name as you suggested, variety 1 and 2 up to variety 100, because 15 you make it doubled as a 100 variety now.
Yeah,
right?
yeah.
So the data set is uploaded. Fine, it is. So variety 1 and 2 are there now. The input data file, it is taken as a variety 1 and 2 are two different varieties.
Yes, yes, you are right. Actually, it should be V1, V1. Like this, you have to be.
V1, V1 only.
Yeah, yeah. So anyway, it will take whatever we give, because here we are seeing the population level, not at individual level.
Yeah, yeah. Okay,
so
sir.
For software, it doesn't matter.
Okay.
Whether you give 10 varieties or 20 or 100, whatever, it will consider as long as it has following that thing. Okay.
Suppose two varieties.
Yeah.
Another variety name is V2.
Yeah.
If we'll give it one for V1 for first two and third and fourth V2, V2, will it take it or not, sir?
First and second V1, V1.
V1, V1. Suppose same variety, no, deployed.
Yeah.
Deployed, you have taken V1, V1. Third and fourth is V2. Fifth and sixth V3, V3. If it's given the same way, the software will take it or not, sir?
I don't know. We have to tinker with the software whether it takes or not. Okay.
Sir.
Yeah.
So now it's since diploid, we have 2 entries for each
Yes.
genotype. So when it's tetraploid or polyploid, then it depends on the number of alleles. The entries will be more for each individual.
For triploids and tetraploids, I don't know how to handle. Maybe you see that manual. If any possibility is there for triploid, maybe 3 alleles we can give or 4 alleles. You can try using that, but I never tried.
Okay, sir.
Okay.
Sir, just a small request from my side, sir. Maybe at the end of the workshop, sir, can you just let us know how, suppose you have done using the agarose electro for the gel, which you had shown in the DARWIN software part. So can you just blend that particular data file in case of how to basically put it in the Excel file, basically, because I have some confusion related to, suppose a particular marker has a number of polymorphism is being shown. So how can I do the entry, sir, basically?
Entry means we have to enter into Excel only. Manually, we have to enter.
Okay, sir.
Or
so suppose
actually,
if I particular
in some of the gel documentation systems like BioRad, they have an option. I think they have a quantity one software where you can assign the allele size for each band. So it will recognize like 100 base pair, 105 base pair. Those things I think you can convert into Excel. Maybe we can try like this.
Okay.
Mahesh. Mahesh.
The same molecular data can be fed to tassel.
This one.
Yes, sir.
Tassel accepts a different format. Same
Different.
yeah, same data you can feed, no problem, but format should change.
Format. So even with the multiple alleles, I can give the same way. V1, V1, V1 like that. I have to give, sir. V1, 1 like that.
V1, V1, V2, V2 like this you have to do. For multiple alleles,
that is better.
I don't know whether structure can handle the multiple alleles or not.
Structure handled, sir, given a population correctly.
Okay, okay. Maybe that we can try. Okay.
Yes, okay.
Okay, shall we move to the next step?
Sir.
Okay, so parameter set, you have to click a new one. Okay, so actually length of burn-in period and length of NCMC repair. Usually for the publications, we have to give at least 1 lakh to 10 lakh length of burning period. Actually, to run the structure software, it takes sometimes, suppose if you have a 200 varieties, some 300 markers, if you give 1 lakh burning period, it will run 2 to 3 days. Or if you have a high performance computer, it may finish within a few hours. Okay, so for the sake of demonstration purpose, I am just giving 1000 only, because it will take a lot of time to run it. Okay, otherwise for the publication purpose, at least 1 lakh. And the ancestry model, use admixture model, because basically it will show you the admixture, whether our population has how much admixture, 50% or 60% or something like that we want to know. Okay, and allele frequency, let it be there, no problem, no need to change anything. Okay, press name the new parameter set, like I'll keep workshop. Okay, parameter setting over. Now you have to start a job. Okay, go to the project, start job, click this one, workshop, and the K is how many populations you are expecting from your data. Okay, so because sometimes as a layman we do not know how many populations are there. In that case, for let us assume minimum 2 populations, so like 10 populations also we can do. But for the demonstration purpose, I'll reduce this. And the number of iterations for publication purpose, it should be at least 5. Okay, otherwise yeah, they will not accept. So, but here I am giving only 2. So later I am assuming.
So for publication case would be, sir, basically
population.
For publications, K, it should be basically, sir, how much?
Yeah, because for publication they won't ask how many you have chosen. Finally, you are going to select only 1K only, because you do not know how many populations are there. You are anticipating that my population has 2 to 5 or my varieties. It may end up with 2 or it may end up with 3 or 4. Now I will explain you how to select the best K. That we have to keep in the publication. Okay. So iteration should be 5 at least. Now the job started. So it will finish in a few minutes, because everything minimum I have given. See here you can see in the under the results, everything it will run. See 2 to 5, everything it will run 2 replications, means 2 iterations. Okay, each run. All are running. Okay, mine is job completed. Press OK. Okay, then we have to find out which one is the best K value. Okay, so here you can see summary, simulation summary, where actually LNPD values will be there. Okay, these values are very, very important. Okay. Can you see my PowerPoint?
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes,
So
sir. Now,
this is the LNPD values we have to consider. From this, we have to find out which will give the best K value. Okay. So there are two methods are there to identify the best K value. One is the use LK, that is the non-parametric test or Wilcoxon test. Here, when you run in a Excel sheet, you just copy paste into a Excel sheet. You make a just graph. So the point where actually the K value approaches plateau, like here you can see these dots, they are not, they are keep increasing, but from here onwards almost like reaching plateau. Okay, so that considered as a K. Whether it is a here, you can see the 5 is the K. Okay, that is the one method. Second one is the delta K. So here, where the maximum value of this what is it? Delta K is there, that you can consider as a K value. Okay, so these are the two methods, and I'll show you. So here you can file, you can save as a text file somewhere maybe in the desktop. Use file permission email. Or you can do manually also. This LNPD values, you just enter into the Excel file. Okay, along with this K2 value, and wherever reaches plateau, we can consider that that as a best rank. Let us see based on this value 1, 2, 2, 3. Let us assume it is K3 is the best one. We select this run 7. Yeah, K3. Then you go to the bar plot, show the bar diagram. So sort by Q. So based on this, it shows the 3 populations are there. Okay. Suppose you want to see at individual level, like each individual has what type of populations, then you can see here by pressing the plot in multiple lines. Okay. So in the y-axis, here you can see the admixture level. Here you can see the first 25th, they has 100% population one type only, 25th, 48 and all this. But if you come here, see the first one, it has some admixture from population 3, some admixture from population 2, and the remaining maximum is population 1 also. So like this we can know. And here also you can see the inferred clusters. So 3 populations made 0.680, 0.165, 0.155. So based on this, this variety belongs to the population 1, 0, because it is showing the more than around 70% belongs to the one population. So it belongs to population 1. So like this we can infer. And this data, this is called Q matrix. So this data you can copy and you can use it in tassel also for running the marker trait association. So that option also there using Q matrix. That's why structure has to run before running the tassel. Or without this also we can run using the kinship values and so, but it depends on. Is it clear? Hello?
Yes, sir.
Everybody got this bar diagram? If you press the 4 K value, it will show the 4 population. Can you see my screen? PowerPoint screen?
Yes, sir. Visible, sir.
So this is the one paper. This picture I got it from recent paper published in last 1 week only. They have done Thai rice population. So they made it into 5 populations. The population 5 is admixtures are very less. Okay, but here you can see the population 2, 50% of one type of population 3 type, and remaining 50% population 2 type, like this. You can. And this is our work using the different gene. This is the SW5 gene. Gene level we have studied population structure in 200 rice variety. So here we could make different populations, different clusters. So like this we can present in the publication. Okay. So this Q matrix values we can export to the tassel to run the tassel software. Okay. So that is about this thing. Any more questions? There are several runs in K3. Which run number to select for Q matrix? So as I told you now. So we have to make a graph using that values, LNPD values. You copy this K1 minus 3488.9 and all these things. You make a simple graph in the Excel sheet. Like this here you can see. Wherever the K values start giving the plateau, that you can consider as a K value, like 5 or 10 or whatever. Okay. Recording of your presentation. I think so far 4 recordings I sent you all. Did you all get those 4 records?
No, sir.
Sir, I had an issue with that thing. The YouTube link which you have sent in the email IDs, sir, whenever I open particular link, it shows video is private, so we can't basically view the file.
No, actually, yesterday morning, I sent a few files which are not opening, but later I sent 3 more. to the participants.
Sir, it's the same, sir. Again, which we have changed recently. I had also gone through that. It's showing the same thing, sir. Video is private.
But others saying they are opening. Madam,
I think you should try to open in your computer, or you should log in with the same email ID which you have registered with the workshop.
I don't know, but others are saying it's opening. Bharat?
This is the problem.
Sir, yes, sir, playing, sir.
They're playing.
Yes, sir, all are playing, sir. All the 4 are playing.
You just try to log in with your username and password which you have given to us during the registration. I think because I shared, it's not public, it's private to the only participant. Okay. What about others?
Sir, in today's email, I did not receive it. Maybe some students were
Sir, I
actually.
did not receive any email today.
Okay, sir. It's opening in laptop, sir. It's not opening in mobile, but it's opening in laptop, sir. I got it.
Yeah, mobile you don't try. Mobile nothing will not work. So those who are facing the problem, I didn't receive any mail in regard to video. Because whatever mails you are given, I'll show you also. I think I already sent you morning one file to check your names and all these things. Did you get it? At least
Yes,
that
sir.
mail?
We got that mail.
File we received regarding videos, we didn't receive any mails.
No, regarding videos also I sent, madam, for all.
Today we didn't receive, sir, regarding video.
Vaibhav video also I sent. Yeah, participants, right?
So we have received it, sir, by ourselves also actually.
You have received?
It is an update, sir. Once if we check the updates, we can see there.
Yeah, it should be there. See, this is the mail IDs I have. Total 269. So this is a to these participants only I am sending. Please check once again. It might be in the spam or you just update it. If still not opening, then we will try to fix it. Don't try to open in the mobile. Okay. Anything, or you can fill the any problems questionnaire. Received only participant list and today's PPT. Then I have received the Word file.
Sir, 9 communication from your side, but today only 2 communication were there.
Yesterday also I sent videos to many people. They said they're opening and they received also like this, they say.
Yesterday received, sir, but not opening.
Yesterday second time I sent, madam, you just check it. But whatever mail IDs you are given in the registration link only I am following. If you have any other mail IDs, you just mail me. I will maybe update.
Sir, also can you please share the PPTs as well along with the videos?
Yeah, definitely.
Okay, sir. Thank
Again, sir, video is not there. Showing Amaravati madam's video only. I will see because I have to test in some other laptop. No mail I have got till now. Tirumala. Okay, videos. I'll try to fix it. See, Anu also first 3 lecture videos are there. Yesterday's videos, but no communication received today.
Yes, sir. Till now, 3 YouTube videos are there, sir.
Yes, 3 morning I sent, and one more I sent yesterday night, I think. Night or today morning, I forgot.
Yesterday, yes, sir. Yesterday we received one email, but that is related to the data required for today's workshop, sir. Like that.
Okay, we'll try to fix. I'll see once again.
Yes, sir. Yes, sir.
From my side, any fault is there, I'll try to rectify it.
Yes,
So
sir. Till now, Uma Maheshwari madam and Dr. Sujan and Amaravati madam, these 3 YouTube videos came, sir, and they were working fine.
But Roja said when you click the Sujan, it is going to Amaravati.
Yes, sir. Same video is coming, sir. I checked.
But how come one person is coming nicely, another person? I don't know. I didn't.
Sir, in the video's name, it is Susan sir's video, but when we are playing it, it is coming as Amravati ma'am's video. That lecture is coming.
Okay, I'll see once again, because I am not a technical person, so I am learning all these things.
Starting.
Yeah.
But after that, only Sujan sir video is there, sir.
Yeah. Roja, actually, Amaravati introduced Sujan, no?
Yes, sir.
So you might have thought that is also Amaravati's task.
Yes, just a little bit go forward, then you can able to hear.
Because that.
Yes, sir, I'll check again.
Please see, some small minor mistakes might be there even from your end also. Don't try to open in the mobile. Okay, try in the laptop.
It's working in mobile also, sir. I am seeing in the mobile only right now.
Okay, but some people says they have some issues. Anyway.
YouTube channel directly.
Yeah. So you please first finalize your name as well as the institution, so that we can prepare the software, certificate. Okay, because that is important for all of you also. If you have any technical issues related to the software, you please use that questionnaire so that I try to answer. Okay. Amaravati?
Sir, Tassel, I am unable to install, sir. Can you send the link for installation, Tassel?
Tassel?
Yes, sir.
I think we already sent in the first mail.
Yes, sir. That's why I could not install through that link, sir.
Okay, I will send once again, no problem.
It's saying that your computer may be harmful. Will you allow to install? So
maybe in that case, you disable your antivirus and load. Sometimes the antivirus may not allow some software. Even Mega also, morning when I tried, it's not allowing to download. In that case, you disable your antivirus so that you can load it. Okay. Any issues, you can mail me or use the questionnaire. So kindly resend the videos. I will try to fix, madam. I'll go through once again. If any fault is there. Okay. Okay.
Okay. Okay, sir. If anything is there, they can send in this that feedback form, sir.
Yeah.
We'll answer. Now it is already time up. Dear participants, your queries will be answered through that feedback form. Okay, and all videos you are going to see. Don't worry. And, thank you very much, sir. Really very elaborative and very nicely explained with a lot of patience. You answered each and everybody's requests. Thank you very much, VLN sir. And all the participants, really, I am very much delighted that you are actively participating. Thank you very much, and we expect the same from all the participants. Thank you, and see you tomorrow, 2 o'clock.
Okay, thank you, Amaravati, and thank you. Have a nice day.
Thank you, sir. Bye, sir.
Okay.
Thank you, sir.
Thank you. Bye.